BioRails, Assay Requesting for any biology, and just a little house. Type questions into the question box if you'd like. So feel free to ask questions along the way or type them in, and we'll address them after the presentation. So without further ado, what I'd like to do is introduce our speaker, who's Dr. Andrew Lemon, the CEO of The Edge, who will give an overview of The Edge as a company and then BioRails as a platform, and also for biological assay management for any type of biological process. Thank you very much.
Thank you, Doug. So, yeah, so today we're going to be talking about the assay requesting features of the BioRails platform, and particularly with a focus on addressing not only small molecule work, but also large molecule work and other types of service that can be requested and tracked. I'm going to be reviewing some of the challenges involved, and then reviewing some of the ways in which our software meets those challenges, and then I'll show you a demonstration of the system we can round up with benefits and questions at the end. So let's get started. So a little bit of an overview of the company very quickly. So we're an independent company. We've been in business for over 10 years. We're based actually in Guildford in the UK, but we have people working out of the US in Boston, and also in the Netherlands and the Czech Republic, and we're serving customers right across from the west coast of the US through Northern America into Europe, and as far out as Japan and Shanghai in China. We specialize in being domain experts in the biopharmaceutical and pharmaceutical research areas, and our software is used by 4 of the top 10 pharmaceutical companies in the world, as well as customers right across those geographic regions I mentioned. Our goal is to deliver solutions for really centered on the biologist and covering everybody who needs to be able to interact with the biologist, and also how the biologist supplies data back to them. So we're covering the full workflow from an assay request and planning procedure through to actually executing and capturing data during various types of study and screening experiments, and also supplying and tracking compound availability and plates and vials during that process. If there's one thing we're passionate about, it's really helping you to optimize your processes. So we really enjoy getting in there and understanding more about how you do your science and the workflows involved, and helping to use our software platform integrated with other systems you have to try and smooth that workflow out and to optimize the efficiency of those processes, actually while keeping the value of the data, which is a key thing for biology. It's really important that we capture not just results, but also the dimensions that they're captured against. So that means the conditions and settings that an assay might be run under. So in today's webinar, the objectives are to review the assay requesting module, and we're really with an application not just across biotherapeutics and small molecule work, as well as other things as well. We're going to review the challenges raised by those different environments and look at our solutions for those, and then I'll give you a demonstration of the system, and finally we'll review the benefits and take your questions. So if you can think up some good questions all the way through, that would be really helpful for the end. So a little about some of the challenges involved with delivering assay requesting systems into a modern drug discovery and development environment. So the first, really, it covers these 4 bullets here. The first one is the diversity of different types of experimentation that are going on in an organization and into the organizations that they're networked with. The second one is the variability of the different types of activity that are going on. And the third one is being able to manage the workflow. That's it, connecting people together and teams and organizations in order to efficiently run various different types of workflow in the organization. And then the fourth one is around tracking and optimizing the overall efficiency of that process. So to drill down into these a little bit, we'll talk about diversity first of all. So obviously traditionally there's been a spread between small molecule research and now large molecule biologics and antibody research work. So we need a system that's capable of managing not just small molecule sample-based requests, but also biologics requests. It could be requests for development of targets into an assay. It could be dosing of an animal or sampling and the analysis of samples. So it's got to be very able to cope with these different environments for requesting services and assays. It needs to cope with both production work but also post-production work as well. So that could be protein production workflows and also post-production characterization assays and services. Obviously it's got to work across discovery and development, so we want to go right down to areas like formulation studies, scale-up work as well. So we're really dealing with lots of different types of entity and different types of research work, because the greatest benefits are really to have one system that could track all the requested activities for organization, and then so that you can start understanding the flow of information and some of the key performance indicators that are going across different activities in an organization. Another area of variability that's involved with those different types. So we're going to cope with everything from early stage assays, where the only thing might be which compounds to test, right through to later stage ones where we're maybe doing much more involved study-based work, where again there may be a variety of different study conditions that are available, and there might be a negotiation that needs to be done between the requester and the expert in the lab that are going to define the study protocol. So here we're talking about everything from screening work right through analytics work into the sort of medium throughput areas where things might be a lot more manual and more complex, and then into the sort of study work, so where again it may be run over multiple months with lots of different subjects and lots of different mini experiments going on within the study. Of course, workflow is also an important feature as well. We need to be able to manage various types of workflow during the process. So there are strategic workflows, which are more to do with the flow of information across different types of experimentation within a project. And then we want to be able to manage those across projects as well in more programs of work. We also want to be able to manage tactical workflows, which may be just the interplay between different organizational units, or indeed out with our contract research organizations and partners that are doing parts of the work, and of course between individual scientists within a workflow. I've got a couple of example workflows here. The first one's a protein production workflow. So here we're going to submit and track the evolution from the initial protein design work right through to the purified product, which is ready for characterization. So we might start with a target product protein that's come from a design, go through expression, which is expresses a protein expression batch, which is then submitted to a purification assay, where it's purified through to a protein purification batch, where it's analyzed with LC-MS. And you might have lots of different activities that go on during the protein production process. It's used to improve the overall protein production efficiency and give transparency across an organization to where the bottlenecks are and where the processes are at. Of course, we want to be able to track and exploit the transformational process right from, maybe the initial gene through to the expressed protein, through to the antibody and into the antibody drug conjugate, and collect alongside all that information also the characterization data that's important to give people that information and know that it's readily available to look at the lineage right back from the from the final antibody drug conjugate through its lineage back to the original genes. Another example of a workflow would be a more study-based workflow, which might be more tactical in nature. So here we're talking about a study that involves lots of different people. It might not just involve scientists within the same organization. It might be that we're doing the bioanalytics, for example, out with a contract research organization. So we've got to go through the process of requesting the overall study, running through the study design, requesting and receiving formulation information for the drug, as well as doing things like method development for the analytical techniques so they're available for bioanalysis and formulation testing. We might want to request animals from an animal welfare group which have been prepared for our study, and then of course we want to go through a set of dosing, sampling activities before we do our bioanalysis and then bring all the data back so that the study report is finally available for release. So there's lots of people involved in this. It's a networked activity, it's team-based, and there's lots of opportunities for chains, weak links in this chain to let inefficiencies creep in. So we're designing a system that can help connect everybody together and make it transparent where you are in that workflow. Another challenge, of course, is tracking. So we want to make sure that we never miss a study. This is particularly acute for some of our contract research customers, where they're receiving requests for studies from multiple clients, often across vast geographies, and they need to make sure that they don't miss those requests and that the service requests and their clients are tracked, and they know exactly where they are and that they're running that process as efficiently as possible. It's designed to eliminate transcription errors, so you're not copy and pasting between different systems, mainly by eliminating the checking procedures that you do by that, so you don't introduce a manual step. We also want to make sure that the next person in the workflow are informed that they need to start their work once the last person's finished, so we can drive out some of these inefficiencies. And of course make sure overall that we're reporting accurate information on the progress right back to the requesters during the delivery of the assay services they've asked for. This is really designed to give managers and lab heads the tools they need to actually manage the processes going on in their laboratory. So a little bit about the solution. So the solution is based on the BioRails platform. BioRails has been around for quite some years. We originally started it back in 2006, and we're currently working on a BioRails 5.1, and 5.0 is in production at a large pharma across multiple sites, and we have lots of customers, of course, on version 4 as well. What it does, it provides a flexible assay request system that can be used for both production workflows, biological assays, analytical requests, formulation requests, even things like synthesis requests when a compound drops below a certain level in stores can be used. In fact, you can create a service that can be requested for pretty much anything using this system. It's very flexible. It allows you to define the input parameters that you want the requester to choose from, and also manage the workflow as you go through that. We can also introduce things like automated requests to cut down on manual tasks. For example, we're working in a program and there's a defined set of assays arranged into tiers. Then obviously every time there's a new batch of material, it clearly has to go into the first tier of assays. So why make scientists make that request manually? It should automatically happen. So we could trigger on the samples from an electronic lab notebook, for example, and automatically submit them to assay queues for processing. And that might be analytical techniques on the samples produced, or it might be characterization of tier 1 assays, for example. The idea here is to make sure that project meetings are more about discussing and progressing materials and making decisions, and less about tracking things because there's a system that takes care of all of that. We also want to be able to handle scheduling activities, so we want to be able to schedule experimentation according to the resources that we have. So that could be people, it could be time, availability. It could even be things like budget and lab space. So if we're externalizing activities out to contract research organizations, we may need to take into account budgets when we do that as well. It's also there to support you with planning as well. So we want to manage the demand coming from project teams and then distribute that work across the delivery teams and external organizations as most appropriate to optimize throughput. So we're going to evaluate and order materials. So it could be, biologic samples coming from the inventory system or small molecule samples coming out of compound management. Then prepare assay-ready plates or near assay-ready plates for the assay labs, and of course generate the information required for execution. So that could be lists of conditions we want the studies run at, or it could be sample lists, it could be other things as well. And then link them to the executional system. Of course, we have a BioRails data management module as well, so you can directly transfer from the planning activities into a real test that's run and the data is analyzed in BioRails itself. Or just as likely, we can link it out to external systems, such as other LIMS, electronic lab notebooks, and data management systems to actually track the execution process. So once we've identified and planned which unit of execution we're going to run, in other words, the experiment, we can then transfer that experiment, including the conditions, over to the execution system. That means that we're going to avoid transcription errors by transferring that data automatically. So basically, we're making sure the execution is then tracked and update the requesters and managers on progress as it goes along, even though it's executed in an externalized system. We can also automate the recording of key performance indicators, so we can track not just the fact that it's running, but also the statuses that it goes through, and also record the times at which those status change happens, so that we can later look at the details of those. In terms of integration, we can then, the system is usually integrated with a number of different systems. For example, it will be integrated with a biologics or small molecule registration system, so we can validate that the samples you're submitting actually exist and in a system, and we can withdraw some of the properties of them as well. It's also integrated, as I mentioned, LIMS and ELNs and data management systems for execution, so we can transfer a unit of work over and then actually record the results in those systems. It's also typically integrated with your ordering systems so that we can evaluate availability and issue orders for material to be delivered to the assay labs ready for execution. We may also integrate it to a data warehouse or data mart so that we can evaluate results once they're posted and then make decisions about where to progress the materials next in the project. All of these integrations are managed using the BioRails Master Data Management Catalog, which allows you to readily integrate those different pieces of information. Once all that's in and integrated, of course, that means that we're capturing real-time information on the delivery process, which means we can take advantage of that information and produce real-time metrics on things like throughput and turnaround times. So this means that we can then dashboard that information to give it to managers. So in this example here, I've got a set of assays from a DMPK department, and we're tracking the turnaround time in days. So, and here we're plotting on a candlestick diagram the maximum turnaround time, the minimum, the median, and then the upper quartile and lower quartile ranges. So that means immediately that we can review the system and we can see, if there's a large range of different values, then we know there's perhaps some process consistency issues going on in there. We might also, if we see that we're not meeting our service level agreements in terms of turnaround time, then it gives us a quick indication of doing that. We can also use these metrics as well to start being more predictive about where we put our resources to resolve and improve and optimize our turnaround times. So that's a quick overview of some of the challenges and some of the goals of the solution. I'm going to move into a demonstration now where I'm going to show you the system. I'm actually going to show you version 4.5. There is a 5 version which is in production at One Pharma but isn't on general release yet. We'll be doing further updates on the 5 platform in future webinars. So in this one I'm going to show you how to make a request. We'll do a simple HTS screening request, and then a more complex study where we're going to set the protocol details as well, which is an in vivo pharmacokinetics assessment. I'll also show you some examples of biologics requests for biologics services. There'll be a biologics formulation, and then maybe some characterization assays as well as a request for a production workflow. I'm then going to show you how we can monitor the overall project itself and start making progression decisions based on the data that's returned. And finally, I'll talk a little bit about managing peak demand and then how to interplace this with managing externalization of studies out to contract research organizational partners. And I'll also talk a little bit about it from the perspective of our contract research organization customers as well, that also use BioRails. So let's, without further ado, let's go to BioRails. So I've set this system up as a fictitious company called LC's Pharma, and we're going to be making some assay requests and tracking them, and also looking at an overall project as well. So I'm going to log in as someone called Walter. Walter is a project leader, so he's responsible basically for making requests on behalf of the project team and tracking the progress of his projects. So we're going to start off by making some requests. So I'm going to make a request. In this case, I'm going to be submitting a set of lots of compound on behalf of a project. In this case, I've configured this system to have project codes which are based on different customers for a CRO. Normally these are basically project codes for the internal pharma if it's a pharma, or if it's a CRO it might be the customer's project codes. All of the customer codes come from usually linked to a system which registers all of the customer projects or the internal projects, and we can link to corporate systems that manage that as well. So we'll do this for customer, let's do it for customer A. And if I want to, I can add a description for the request. And then basically in here I can paste in a list of lots, or I can import them from a file, or if I want to, I can just find them by looking them up. So here I'm just going to add, we'll just do a small one in this case. We'll add a couple of lots to the system. You can see it checks to see if I've already added it, so you don't accidentally add too many or add the same one twice. So add a couple of those. And then we're going to come in here and we're going to order some assays. So underneath here I've basically got to look up with assays available on the system, which are available to me, Walter Skinner. There may be other assays in the system which are not available to Walter, so immediately I can see down here, I can see these grey one here, I'm able to review them, but I'm not able to submit to these particular assays. So you can see there are various different assays here. We've got some ADMET assays, so permeability assay for CAC02. I've got some pre-registration assays for log D purity and solubility. I also have a arrhythmia assessment assay, so it's an in vivo pharmacology one for cardiovascular, and a general pharmacology service as well. So this will be an example of a more involved study-based request, so we'll add one of those. And then I'm going to pick a more screening-based one, which is a microsome stability assessment. And of course, if I had access to submit to them, I could submit an HTS one as well, either single point or dose response there too. We also can request panels of assays as well. So if I look to, say, an early ADME panel, just click that, I could add a whole series of different assays at once. So here I'm doing log D, CAC02, and solubility in a single click. Or alternatively, I can select a whole cascade of different assays. So here we can see I'm going to do 5H2 2A, 2A and 2B targets, single point. And then if the results are good, we're going to do some follow-up dose response work. So that's what we call a cascade. So in this one here, I'm actually going to do the two I's mentioned, which I'm going to do on microsome stability, and then also a general pharmacology service. I've configured this system to collect certain inputs from me as a requester, so I can decide details of the protocol for the study. So in this case, a microsome stability, I'm only offering mouse, so the only decision is do I want it or not. If I want to, I could make an individual comment on a particular sample. So I could say this one has solubility issues. So you might want to look out for those. And underneath here, this is a more study-based request. So here I can specify which species and strain. If I double-click on those, it activates a data lookup where I've got a hierarchical dictionary of different species and strains that are available for general pharmacology. I can also specify which gender of animal I want, maybe the route of administration. So let's change this one to IV. And I want the same thing down the whole study. And I could vary the dose, which vehicle is being used, the administration volume, the volume to be ordered, the administration volume, and the number of times. And as the person who runs this assay, you can choose which of these parameters you would like the requester to choose from, or which of them are fixed in the assay. And you just expose those you want them to give them a decision on it. Some studies are more of a discussion point, so you may just want to register the fact you want a study, and then it might be more of a meeting and an email conversation to set the details of the protocol. The other thing to remember here is just because the requesters said they want a dose of 10 mg per kilogram IV, that doesn't mean it's a sensible dose for a rat. So I would say normally it's around 5 mg per kilogram. So either the requester or the tester can modify the conditions as they come through in their study. So let's click next. And then here you can see it's done a duplicate check and it's told me actually I've submitted that particular compound to 3 previous times. So do you really want it again? If you do, you can just say yes. Alternatively, you want to triage. So you might want to say, actually, I don't need all 3 of these against both studies. I'll only have 2 of them. So you can uncheck some of the things you don't want and then confirm the request, and that goes into the system. And what I'll get is I'll get a status of each compound against each requested assay in my request dashboard here, and I can track those as I go through. I can also ask them to send me an email when the status changes as well. So there I showed you an example of doing a request for both a more high throughput study where the only thing to specify was which compounds, and also a more study-based request where I can interact and define details of the protocol. I'm now going to show you an example of a biologics request. So here you'll notice last time I picked a lot of compound, and now I'm going to pick a biologic. You'll also notice that I can order animals through here. So asking for an animal to be ordered and finding details of which species and how many animals. I can ask for a particular animal to be dosed by submitting the animal identifier and asking for a dosing. I could also ask for a biosample to be analyzed, a biologics material to be formulated or characterized. I could ask for a container to be shipped somewhere, a target to be validated, tubes to be shipped, or an siRNA sample to be screened. So in this case, what I'm going to do, and the list of those depends on which services you configured. The system's entirely configurable, so if you're only offering small molecule lot-based services, the only thing in the list is going to be is going to be lots. If you've got a mixture of biologics and lots, you just have the 2 in there as well. So what we're going to do is submit a set of biologics. So we'll say please characterize, and I'm going to submit them again on behalf of a particular project. And I might want to say when I'd like the results by. So I'd like these by the 15th of October. And then I'm going to paste in a list of different biologics lots. So I've got a list here that I've pre-prepared. So I'll just paste those in and then add them to the list. So here you can paste in obviously a large list. If you've got a very large file, you can just import them from a CSV file, or again, you can go and look them up. So we'll click next. And these are all the services that I can submit a biologic to. So you can see I've got some services that are for formulating biologics. So I can do a differential scanning calorimetry screen. I can also do a freeze thaw stability assessment, or this thermolysin stability test. If you hover over, it will show you details of the test, which the tester has provided. You can also see I've got some characterization assays. So I've got some biologics-based in vivo PK assessments, and also an antibody drug conjugate warhead release assay that I've got here. If I want to, I could ask for a whole formulation panel, so just one click and get all the services, or I can select individual ones. So let's have a mouse PK and maybe this antibody drug conjugate warhead assay as well. So we'll click next. And again, there, if there's any details of the protocol for these assays that needs to be provided, it's going to show me that configurable form which the tester has set up. In this case, all I'm doing is submitting the biologics batch to it. So we'll click next. And you can see it's done a check. It said these have been requested before, so I won't do them again. I'll just confirm that request. And there we are. We can see the status of these in these different assays in here immediately. Okay, so the next thing I'm going to do is I've submitted my assays. I'm actually going to do a request this time that's going to be to submit it to the whole project cascade. So create a request. We're going to do a biologics request this time for protein production, which I think actually is a biosample request. So we'll just select which project that's on behalf of. Click next. We'll add some biosamples. So I don't remember them offhand. So these basically could represent a particular design pattern for a protein or an antibody, or they might be a gene identifier that we're going to ask for protein production on. And you can see here I've got a whole cascade of protein generation workflow. So we click on that, you can see it's going to go through initialization, where we'll do our protein generation approval process, which basically is just checking that the thing I'm asking for is correct and filling out the protocol. And then if it's antibody, we'll go into antibody characterization, generation and characterization first of all. If it was a protein, then I'm going to go straight to protein production and skip those other things. Otherwise, I might go through a vector design and generation or a cell line development exercise. And then finally, once I've expressed my protein, I can go to purification step, and then finally the distribution step, where it's going to be distributed to the groups that need to use it. So that's how you would do a whole cascade of them, or I could do an individual one. So let's put it into an antibody generation characterization assay. So that's now going to ask me for any details of that service that I need to fill out. So it might be asking for things like whether it's an antibody engineering, or so it needs to be engineered, or it's a new antibody, or it's a non-antibody protein. And if they're all the same, I can fill those out. If I've got UniProt ID, I can fill that out here as well. I might have a protein sequence I want to give it, and you can see across here all the other information that characterizes that request. So maybe it's the amount that I've got. So let's say I've got 10 milligrams. And a volume. This is the isotype for the species, so it's a rat. Binding profile, and again I can fill out all the details I want on this particular request. Click Next. And then obviously again it does a check to see if I've asked for it before, and then I can submit that into that particular service. Right, so having submitted it, what I'll do now is I've got a different example here where I've submitted a compound to a particular lead optimization program, which is on this particular system here. So this is a PIN lead optimization program. It's a 3-tier cascade. You can see the first tier is a set of chemical property evaluations, so log D, purity and solubility. And you can see there are 693 samples that have been submitted to this cascade. 543 of them are awaiting action. 80 have been evaluated and passed through to the next tier, and 70 are left behind, failed the criteria. And then here you can see in the second tier of this cascade, we've got 80 compounds in it, and we're running a battery of single point assays against various 5-HT targets. And you can see 10 of them have passed the criteria and moved through to a confirmation dose response assay. If we select the individual tiers of the cascade, then we can evaluate the results. So you can see some of these have been submitted and the assays haven't been run yet, so there are no results. We filter them by D values. You can see these first ones here have been tested. Some of them have results that are published, so we might want to filter this by published results. And you can see in here that I'm evaluating the log D at 3 different pHs, the purity and the solubility. And you can see on the right-hand side here there is an outcome. So these have passed. There's 2 ways to pass compounds forward. You can manually select them in here and select them to be passed, or they can automatically be evaluated using something called a progression rule. If I select at the top here, you can see we provide this very simple to fill out progression rule language where you can enter in the rule that you want to be applied. So in this example here, we want to make sure there are results for all of the log D measurements have been made, and then we're going to check the purity is above 60% and the solubility is above 40 solubility units. And if that's true, then it's going to pass automatically. So you can see some of them passed and some of them reached the fail criteria. There's an interface here where you can edit the rules, you can modify them. You must save them with an audit rule so that there's an audit trail. If things pass this particular criteria, then they'll move forward to the second tier. So here you can see we're generating, running a bank of different single point screens, and then we have our inhibition values in the middle. And again, here we've got a rule that says if any of the compounds exhibit inhibition against one target, then I want them to go forward for a confirmation dose response. And again, here you can use and or logic. You can calculate ratios of different ones, take logs and other mathematical transformations of the results in order to make decisions. So if they pass this one, then they'll go forward for a dose response curve. And finally, we're looking for selectivity against 5-HT2B in this example. And if it is, then we're going to pass it and then actually send it to another assay, which might be a secondary cascade, or it could be another assay. So that's the cascading feature, how to progress compounds and review the results. And again, you can use this for small molecule programs, large molecule programs, and other types of workflow as well. Okay, let's go quickly remind ourselves. So what I've shown you so far is how to make a simple HTS screen. I showed you how to make a more involved assay. In this case, we used a general pharmacology service, but it could have been in vivo PK. I also showed you how to use it for large molecule requests, so submitting biologics for formulation or biologics for a protein production workflow. So we're submitting a design for a protein and then running it through the production workflow. I also showed you how to submit biologics to characterization assays as well. I also showed you how to monitor and run a project to make progression decisions. And finally, what we're going to do is look at how to use the system to manage peak demand. So for this one, I'm going to log out of as the requester. I'm going to log in as a person called Barbara Bryson, who is an assay scientist. So we're seeing the other side of the process. So we're seeing all the requesting. Now we're going to look at how to manage the demand coming in and optimize those processes. So here you can see Barbara is a lab manager. She's actually from the DMPK team, and she manages these 3 assays here. So if we can go in and look at the details of the assays, or I can review all of the queues that either Barbara runs, which are the my queues, or the ones for her teams, which are the team-based ones. So you can see here there's everything from blood plasma ratio assay through things like CACO2, PK assessments, and even a bioanalysis study for running tissue samples that have come from development projects. For each one of the assays that we're managing, you can see the workload coming in. That's the pending requests. So you can see there are 79 samples pending for the microenzyme stability queue, and there are 16 being worked on. If you want to drill in, you can drill into the details of the microenzyme stability queue. Just clicking on the link there, that will show me all of the samples that have been requested for my assay. I can see which projects or customer they're coming from. If I want to run them, I simply select the ones I want to run. I can evaluate and look at which conditions they want to be run at. So these are all mouse microsomes. And maybe I want to run these ones as well. You can also see how long they've been sat in the queue. I'm going to put them in my assay cart. And then a couple of things I want to show you. You can see what it's done is evaluate the availability of compound from the inventory system and identified containers which have enough material in to make up the assay-ready plate. So to do that, it's using something called an order profile for the queue. So if we go in here and look at details of this queue, you can see it has something called an order profile. What the order profile does, it says, actually, I need an assay plate which looks like this. It's a dose response plate in this example. This tells you the assay plate format, so I could select different assay plate formats, make those available. It's coming out of a liquid store. It's a Texel, Hamilton Texel liquid store. I'm looking for containers that have 10 microliters of 10 micromolar stock in the liquid store. I'm looking for compounds made up in 100% DMSO. I'm looking for 1 assay plate. And also I can say which controls and standards I'd like to be added and where they're positioned on the plate as well. So based on this order profile, we can submit a request for the assay-ready plate to be prepared from a set of tubes. So if we look here, you can see it's found 5 of those samples have tubes and 1 of them doesn't. So for the one that doesn't, I could go ahead and order powdered sample to be made up in liquid sample and put in that tube store ready for the assay. In this case, what I'll do actually is deselect this one. So I don't want that particular one. And then I'm going to create a task from the results. So create a task. What I'm going to do here is I've actually got too much work to handle for my internal DMPK team. So I can outsource this one to various different CROs, or I can run it internally with my teams. So if I'm running it internally, I'll run this microsome stability results workflow, which not only includes the details of the assay, and but also a template for actually doing the data analysis as well. Or I could externalize it to a CRO. So in this case, I could send it to Aptuit for them to run it, or I could send it to Evotec for them to run it. So let's send it to Aptuit. I could also, if I want to, I could set up the CRO's user in the system with an email address, And then when I allocated them the study, it could automatically email the CRO details of the study that it wants to run. That doesn't mean they have to have access to the system. It would just send them an email notifying them there is a study coming their way. In this case, I'm going to keep it as Barbara. I can also give it scheduling information. So I want them to start on the 28th, and they should be finished by the 2nd of October. And then we'll run that study. What that does then is to create an output process with details of the experiment that I could either run directly in BioRails, or I could pass off to an ELN, a LIMS system, or in this case send it out to the actual contract research organization itself. If you want to get data back out, you can also, there is import and export processes to export these to Excel files that you can send to the CRO to be completed. Then once you get the data back, you can import them to bring in the results. Also, we can pass this unit of execution, this experiment, off to an ELN, like we've done it with various different types of ELN, the BioVia workbook. We've done it with the e-workbook. We've also done it with e-notebook and other ELNs as well. And in those cases, what it does is pass the sample numbers, the conditions that we will run at, and allow them to run their protocol to generate and collect the results and pass them back. So obviously, once I've completed this, I might upload some results to it. So let's just fill in some intrinsic clearances. I'll just put some random numbers in it just so that we've got some results in it, and maybe a predicted clearance. I could give it a scale clearance. I could give it a characterization of those, so we'll say they're all high, and any comments I wanted to fill out. And then all we're going to do is change the status of this experiment to say it's published, which means the results are available. If I then went back to Walter's original request, he'll see that these particular samples in this particular assay are now published and the results are available. Also, we're recording the timings at which you do all those things so we can generate key performance indicators and provide reports to management as well. So what I just showed you was how to manage the microsome stability queue, which we're seeing here. I can manage it. I can assess the workload, and what I've done actually is to move some of the workload out to a contract research organization and then dispatch them the information about what we want tested, and then when we get the results back we can upload them and complete the test. So that's showing distribution of work across internal teams and also out with the contract research organization. Okay, let's conclude this. And of course that works not only with small molecule studies but also with biologic studies, with formulation studies. It could be a formulation study, it could be a scale-up study, it can be all sorts of different services it can work from, right across research and development activities. So some of the benefits of the system, by making these key performance indicators and other things transparent, means you can reduce your cycle times and increase the efficiency of the processes, both preclinical and also some of the development processes as well. It helps you to increase your productivity by delivering highly configurable workflows and analytics to your fingertips, so that you've got the tools you need to be able to manage your teams and contract research organizations. It also simplifies tracking, gets you away from tracking things with Excel sheets and emails, so that it's all managed in one system and transparent to everyone. It makes sure nothing falls through the cracks. So whether you're a contract research organization trying to ensure that you're tested everything the clients have asked you for, or you're a CRO, you're a pharma or biopharma trying to externalize the work and make sure you're getting your results back, it's all tracked and modified and managed inside the system. So who uses this? So we have a variety of different customers ranging from large pharma. So AbbVie have been a long-term customer of our system. They're using it right across, I think it's about 6 sites. There's about almost 1,000 scientists, I think, using the system across all their research activities. Sanofi also use this for analytical requesting and tracking. We have various middle-sized pharma right across the globe. One of the notable ones is Cubist Pharmaceuticals. Very anti-infective. They've been recently acquired by Merck. We'll continue to use the system across various research and development activities. Also, Evotec, so one of our contract research organizations using the system. We've got some others working on at the moment. They use it for registering DMPK services from their outsourced discovery services and also from customer requests. And also Zoetis, which is the old Pfizer Animal Health, they use it for assay requests and inventory work as well. It's basically used to centralize all of the requests so that you get away from email tracking and Excel sheets and centralize it to one system in the middle that everyone can see. Our future plans, we're working on version 5, which actually is in production at one large pharma customer at the moment across sites in the US and Europe. It's a new user interface. There is support for assay planning, so things like consolidation of plates, definition and creation of experimentation files for externalization, and also for passing to execution systems. We're also managing the biologics production workflow there with integration to other biologics production systems for tracking the overall progress and evolution of samples from gene through to drug. We're also, there is a new KPI metrics module in there for a collection of KPI metrics and reporting. Really, this, I'm trying to get across, this is a very flexible solution. It's perfect for both contract research organizations, pharma and biopharma companies for tracking their processes. So with that, that reaches the end of the demonstration part of this, and I'll hand over to Doug to take any questions. Doug?
Hello. Hi. It turns out I was muted within the meeting, but not on my phone. So apologies for that. If there's any questions, please add them into the chat box. I'm also unmuted anybody if anybody would like to speak directly. Question, Andrew, maybe if you can just briefly highlight the licensing for BioRails and Morphit.
Sure, yeah. So most of what I've showed you today has been a module called BioRails PTO, which is the Assay Requesting module. That's a user-based license. You can license it perpetually or annually with maintenance. So, and that covers all of the requesting, also the queue management and planning activities. They're all covered by that license. In BioRails 5, we also have an assay planning module as well, which is used for ordering plates and also plate consolidation, as well as generating all the execution materials ready for your assays themselves. So it will generate sample files, plate maps, and other things that you can use to execute your assay. Of course, there's also modules for actually doing the data management. And as well as that, we have Morphit, which is an analytics tool, which is used to can be used to generate those KPI metrics reports that I showed you, as well as generating things like, well, for CROs, they can use it to generate invoices for services and other things. Is there any questions? We've unmuted participants, so if anybody actually wants to ask a question live, they're more than welcome.
So for the sake of time, I don't necessarily see any, but what I'd like to do is let you know that this meeting is being recorded and we'll send out the webinar to everybody who participated. And Andrew, if you can go to the contact page, if anybody has further questions, then we can also be contacted directly through the website or each of us individually. If there's no further questions, I'd like to thank you all for your attendance. It actually turns out we're right at the hour, and so I appreciate everybody's attendance and participation. Look for these webinars we've been doing on a monthly basis on various topics, so if there's also any topic that is of particular interest, we would welcome your suggestions and input. Thank you very much and have a great day. Cheers.
Thank you.